Hello and welcome back. This mini lesson for chapter 14 is covering the ventricles that we saw developed in embryology at the end of the last video, and then taking a look at the meninges, as well as we'll mention the blood-brain barrier. So all of these are kind of collectively the protective mechanisms that the brain has to protect all of that neural tissue that's super important. All right, so here is a diagram of the ventricles of the brain. So at the end of the first or the last video with the embryology showing the neural tube, it had the hole in the middle. That hole develops into these very particularly shaped spaces inside of the brain, which we call ventricles. Now, if you have any background with heart anatomy, you know there's a left and right ventricle in the heart. It's not the same. It's just the usage of the word ventricle means hollow space. And so we applied that to heart anatomy, and we'll apply it to also the brain anatomy. These are fluid-filled spaces that developed from that hollow tube, opening in that neural tube that developed and changed as the brain structure was developing and changing. There are 4 ventricles. The first 2 are not called first and second, like the third and the fourth are. They are just called lateral ventricles. They're symmetrical. They kind of sit off center in the kind of area of the cerebrum, and they kind of curve around like ram's horns, if you will. So we can see that here, kind of that pattern, and we'll see that pattern actually repeated in some of the other structures in the cerebrum and the limbic system. So their 2 lateral ventricles are the most superior, and then they kind of sit off to the side, and they are continuous with the third ventricle via what's called the interventricular foramen. So again, we're using the word foramen because we know that means whole from our bone list, and inter meaning between. So interventricular foramen means the hole between the ventricles. So that makes sense. So the lateral ventricles drain into the third ventricle, which is more centrally located, and it's actually, the third ventricle is made by the 2 thalamic lobes, the diencephalon, and so there's this hollow space in the middle that's kind of sandwiched on both sides by the diencephalon, the thalamic part of the diencephalon. So that's the third ventricle, and then there's a skinny little connector that connects third to fourth. So here's fourth, so this little skinny connector is called the cerebral aqueduct. It also has some other names, so you might see it to be called the mesencephalic aqueduct, the aqueduct of Sylvie or something like that. So cerebral aqueduct is probably the easiest way to remember that structure. And then that leads down into this fourth ventricle here, which would be continuous with the central canal of the spinal cord. So you can see all of these open spaces are connected to each other. They're filled with cerebral spinal fluid, and it flows and circulates through all of the ventricles and gets recycled, and that's what we're going to talk about in a little bit in a couple slides later when we talk about the production and the circulation of cerebral spinal fluid. So these are the ventricles. And then here's a shot of that lateral fissure that I was mentioning earlier. So your 2 cerebral hemispheres are kind of sitting like this, separated by this big, thick lateral fissure, or longitudinal fissure, sorry. Okay, so those are the ventricles of the brain. Next, we're going to take a look at the meninges. So we have some familiarity with the meninges already because we covered that in chapter 13. So if you'll remember, we have dura mater, arachnoid mater, and pia mater. It's the same terminology that we're using with the brain. We just have to add a few things that are specific to the brain itself, which makes sense. So let's see, what do I want to tackle first? The dura mater. Okay, well, here's the dura mater, here's the arachnoid mater, and here's the pia mater, right? So we're identifying all 3 of those. The pia mater sits right on the surface of the brain, the neural tissue. The arachnoid mater has all these webs, right, and the subarachnoid space. And then the dura mater, this is where we're going to see one of our first differences. So superior, in kind of the superior region of the brain and in some of these other places, that dura mater actually splits and separates into 2 layers of dura mater. We have what's called the periosteal layer and the meningeal layer. Periosteal because it's connected to the periosteum of the skull bone, and meningeal because it's closer to the other meninges on the, like the arachnoid mater side. Inside this split, we now have created a new space, and this is called the dural sinus. The dural sinus is filled with blood. Okay, specifically venous blood, the deoxygenated blood that's going to be heading back to the heart. So wherever you have the split of the dura mater into the periosteal and meningeal layers, filled with that is going to be this blood, and that's going to be part of the drainage of the blood flow from the brain. So we can see quite a few sinuses here. We see the superior sagittal sinus, the inferior sagittal sinus. This is cut off by my head, but that's the transverse sinus. Those are the major ones. So superior, inferior, and transverse sinuses. So that's one of our first differences between spinal and cranial meninges is the splitting of the dura mater to produce these dural sinuses, which are filled with blood. Okay. The other difference that we can see is the production of what we call dural folds. Let's see if I can draw this. I usually draw this on the board, but I don't have as much space. So if we're going to be looking straight on the hemispheres, right? So we'll do my hemispheres. So these are my cerebrum and then the other parts of the brain, right? So if we're going to take our dura mater, right, and then we have our little dural sinus, there are extensions of that meningeal layer that are going to kind of go deep into those fissures like that. So this is what we call a dural fold. And there's 3 of them that we have identified in this book. So the big one that I just kind of drew that goes like this and folds into underneath the sinus and folds into that lateral or the longitudinal fissure is what we call the falx cerebri. Okay. Then we have one that kind of comes in between the 2 cerebellar hemispheres back here. And this is what we call the falx cerebelli. So again, it's just extensions of the dura mater, specifically that meningeal layer of the dura mater comes in and creates this kind of a wedge of dura mater tissue. And then we have another one called the tentorium cerebelli, sits inferior to the cerebellum. So these dural folds help to stabilize the brain. So that is a protective measure. So not only do the meninges help to protect and surround the brain tissue and protect it from infection and some jostling, we have these dural folds to physically hold the brain in place so it's not flopping around inside your hollow skull, which is good. Okay. All right. Covered that. All right, so now what we're going to do is we're going to take a look at the production and circulation of the cerebrospinal fluid. It is also part of the protective properties of the brain because the CSF is found inside the brain in those ventricles, but it is also found outside of the brain in that subarachnoid space, and it actually helps the brain be buoyant. It's like cushioning, kind of shock absorber and cushioning and buoyancy. So all of those things lead to better protection of our brain inside our bony skull. So there are a few places in your brain where your CSF is produced, and these are called choroid plexus, and I'm going to identify that on the bigger picture on the next slide. But this is showing you a super zoomed in region of the choroid plexus. Choroid plexuses are collections of capillaries, a whole bunch of blood capillaries, covered by ependymal cells. So if you guys remember ependymal cells from our chapter 12, they are held together by tight junctions. Right, so tight junctions, remember from 231, don't allow things to go through. Anything that's transferring has to come through the cells, the endothelial cells lining the capillary and the ependymal cells. So this is where CSF comes from. It is a filtering of the blood that's traveling through the choroid plexus capillaries that then become the CSF. So it's like filtered blood plasma, you could say. And so that filtering happens at these choroid plexus locations, and that is your cerebrospinal fluid, in this particular case in the 3rd ventricle. And all of your ventricles are lined by those ependymal cells. So if you remember, that was their function. Lining the ventricles helps to regulate and circulate CSF. They're ciliated, so that kind of sets up this current, this circulation of that flow. Right, so it allows for the production of the CSF. We're going to talk about the recycling of that in a different picture. So this is where it comes from. So then if we take a look at this larger scale picture, we have a couple locations of where we find the choroid plexus. So I'm going to highlight it here in the third ventricle, and I'm going to highlight it here in the fourth ventricle. So those are the two places. So in that previous slide, zoomed in, blood capillaries, filtering out the blood plasma, producing the CSF. This is where it comes from. And so then you can see on this diagram, all the little red arrows, that's tracking the flow of the CSF. So it's produced within the brain ventricles, right? The third and the fourth ventricle. It can flow up into the lateral ventricles. It can go into the third ventricle via those interventricular foramen. It goes down via the cerebral aqueduct into the fourth ventricle. Fourth ventricle is continuous with the central canal of the spinal cord. So we have all of the CSF going through all of the spaces within the brain tissue, right? Lateral ventricles, third ventricle, fourth ventricle, central canal. How does it get into the subarachnoid space? Well, there are two apertures, okay? Well, there's three apertures. There's two lateral apertures and one median aperture at the back wall of the fourth ventricle. And so that allows the CSF to leave the fourth ventricle and enter the subarachnoid space. Okay? So that's all this kind of the step 3 box talks about how my cats are playing, distracting me. So that shows you where and how the CSF leaves the ventricle system and enters the subarachnoid space. Okay, so now it's circulating inside and it's circulating outside, and we saw where it's produced, but where does it go away from? Because we don't want to blow up into this big, water balloon full of CSF in our brain. We can't because our skull won't let it. But where does the CSF go for continually producing? And we do about 0.5 liter a day. So about 500 mils of new CSF is produced every day. So we have to recycle about 0.5 liter or 500 mils. How do we do that and where do we do that? So here is your, I'm kind of highlighting, this is the subarachnoid space, right? All of the spider webby looking stuff, that lets us know we're in the subarachnoid space. That's where the CSF is flowing. In those dural sinuses, specifically they're showing the superior sagittal sinus, kind of up here, we have these little, there's a couple names for them. They can be called an arachnoid granulation, or they can also be called an arachnoid villus. I think that's what your lab manual calls it. So either a villus or a granulation. It's these little pegs of tissue that penetrates through the meningeal layer into that dural sinus. The superior sagittal sinus in this case. So all of that CSF flows out into the blood. So that's the drainage, if you will, of the CSF, and then the superior sagittal sinus, since it's full of venous blood, that drains out of the brain back towards the heart for recycling and cleansing through the lungs, getting the oxygen, dropping off CO2. The kidneys filtering out wastes, your spleen, all the filtering structures within your body are going to be taking that blood, and then the new fresh blood is circulating around, and the new fresh CSF is produced in your choroid plexus, and then the old CSF is recycled at these arachnoid granulations in the dural folds in the superior sagittal sinus, right? So there's lots of parts and pieces to this process. So make sure as you're learning the cerebrospinal fluid circulation, you become comfortable with all of that terminology to help describe the flow of that. If there is a problem somewhere in either the production of the CSF, the flow of the CSF, or the recycling of the CSF, you could lead to a buildup of fluid in the brain, and this is usually called hydrocephalus. I think there's a little clinical section in the textbook. You can read through that. Yeah, so that could be a problem, because if you have a whole bunch of fluid pushing on your neural tissue, it's not going to allow your neural tissue to do its job, which could lead to symptoms or malformed brains if it's happening in a baby that hasn't formed their brain fully yet. Okay, so that is your production and circulation of your cerebrospinal fluid. The last thing we want to talk about in this protection of the brain lesson is the blood-brain barrier. So the blood-brain barrier pretty much is just talking about tight junctions in the endothelial cells and the astrocytes. surrounding the capillaries in your brain. So if you need to go back to review from chapter 12, glial cells, so if you remember, the astrocytes were the most numerous of our neuroglia of the brain. And there's a good picture in your chapter 12 that showed astrocyte projections completely surrounding a blood capillary. So there's nothing going to be leaking out of that blood capillary that isn't allowed to come out of that blood capillary. So there's going to be tight junctions, they're kind of showing them here, tight junctions between the endothelial cells, and there will also be tight junctions or something similar to them between the astrocyte projections. So if you are a substance, green circle, and you want to leave the capillary to get into the brain, you probably can't. You have to have certain characteristics because you'd have to go through the plasma membrane of the endothelial cell twice, and you'd have to go through the plasma membrane of the astrocyte cell twice. So that's 4 plasma membranes you're going to have to get through to be able to make it out into the space where the neurons and other neuroglia are. So what are some of those characteristics? Well, you got to think back to your understanding of phospholipid bilayers and what is naturally permeable to those phospholipid bilayers. So I have my little stop sign guy, a comic in the corner saying, hey, we want in. We're little particles that are in the capillary. Can we come in? He's like, no, I'm sorry, but you're charged, you're big, and you're not lipid soluble. So you cannot enter the brain. So those things are going to get blocked. So what does that mean for the things that can get in? They have to be neutral. So no charge. They have to be small, and they have to be hydrophobic. Remember, hydrophobic is the same thing as lipid soluble. So if you are neutral in charge, you are small in size, and you're lipid soluble or hydrophobic, you have the ability to freely diffuse through these plasma membranes and you can leave. However, if you're charged, if you're large in size and you're hydrophilic, then you cannot just leave. You'd have to have an ion channel. So maybe things like glucose, because glucose is charged, it's pretty big, and it's hydrophilic. So you would, if you're glucose, my little blue circle here, you would have to have channels in all of those membranes to be transported, probably a facilitated diffusion, through those membrane channels to be able to get into the brain. And so all of those endothelial cells and those astrocyte projections, they all have specific facilitated diffusion channels that will allow the things to come into the brain that need to, and let everything else just stay in the blood supply and it can't get into the brain. And so that's very important because it helps to regulate the environment around the neurons, because as you learned back in chapter 12, sodium concentrations, calcium concentrations, potassium concentrations, neurotransmitter concentrations, enzyme concentrations, all of those have to be tightly regulated to make sure all of the neurons are functioning just right. So that wraps up. Oh, sorry, I'm looking in the notes. I forgot this last piece. There are 4 places in the brain where the blood-brain barrier is not as strictly regulatory as it is everywhere else in the brain. So there's portions of the hypothalamus, capillaries in the pituitary gland, capillaries in the pineal gland, and capillaries in the choroid plexus. So the first 3 things are all related to endocrine function. So that's where hormones have to come into and out of. So you need to be a little bit more liberal with what can enter and leave the brain when you're dealing with hormones. And then the choroid plexus is where CSF is produced. So it's not quite as restrictive in the production of CSF as it is in just the, say, a capillary in your temporal lobe or something. All right, so that wraps up our discussion on the safety and protective measures around the brain. I will see you next time. Bye.